Okay, everyone, welcome to the evolution quiz review. So feel free to ask any questions that you've got. Sammy, they can't see you. Please say hi.
Hello.
Say hi.
I have yummy cookie.
Okay, we'll eat them. We'll offer them cookie later. Okay. Okay, so as a review, the quiz will cover from pages 83 to 94. All the topics of evolution. So starting from population genetics, you should be able to calculate allelic and genotype frequencies. You should be able to do chi-squared. You should be able to figure out if a population is in Hardy-Weinberg equilibrium.
Hello again.
No, Sammy, please go.
Hello again.
Turtle. Okay.
I have a hand.
Is this your pink turtle? She's asking a question.
Yes.
Okay. Let's see. And then, of course, the topics of evolution, speciation, ending with classification, origin of life, etc. Remember, there are practice quizzes online, and ideally by the end of tomorrow, you need to finish problem set 6B, and then I will post the answer keys to those to help you study. So go ahead and ask any questions you've got, and I'll just go from there. Let's see. Until people do ask questions, let me see, what can I talk about? So we'll just start from the beginning. I'll just start reviewing the topics, and then you guys can ask questions as you have them. For allelic and genotype frequencies, remember the calculations shouldn't be too complicated, but one, you are going to want to have a calculator for your quiz. And then also remember that there is a very big difference between expected and actual values, right? Expected values are always based on probability, or they're based on Hardy-Weinberg equations, whereas observed is going to be, well, if I have 10 tasters out of 100 total students, right, then I have just a percentage of the total. I mean, as long as you could probably round about 2 decimal places, and that'll be fine. Even if your numbers end up being a little bit off, it'll be fine because I can still tell you're in the right ballpark. Whereas if your numbers are way, way off, then, obviously that will tell me that you didn't quite use the right numbers or something. Oh, thank you, Gina. Okay, thank you. Okay, so anyone? Questions? Let me see. Remember, for evolution, you don't need to know the specific examples from the lecture, but you do want to be able to take any example and apply it to various terms. Ideas of like natural selection or artificial selection, or the different types of selection, being able to apply which one it is, and that type of thing. Let's see, what else? Let me pull up the practice quiz then and see what people might have questions about there. But feel free to ask quizzes ask questions as I'm doing this, since that's always going to probably be the most useful part for you guys. Let's see. Bless you.
Thank you, Mom.
Oh, still no questions. Okay. Well, let's see. Oh, okay. There's some interest. So your practice quiz has a lot of like Hardy-Weinberg stuff. You won't see that much of it on your actual quiz. This was just to give you a lot of different practice. So just be aware of that the percentage of Hardy-Weinberg stuff you see on the practice quiz is not indicative of the amount of Hardy-Weinberg stuff you'll see on your actual quiz. But as long as no one's asking questions, I guess we will start there. So remember, Hardy and Weinberg, their model describes a not evolving population. So they had two variables, P and Q, where P is the allelic frequency of one allele and Q is the allelic frequency of the other allele. Now, really can go either way, but by convention, usually P is the allelic frequency of the dominant allele and Q is usually the allelic frequency of the recessive allele. What Hardy and Weinberg said was if you know P and Q, if you know the percentage of each allele in the population, then you could predict, assuming it's not changing, the percentage of individuals who would have each genotype. Where the genotype frequency then of the homozygous dominance would be Q squared. Oh, Sammy, can we not do that right now? It's very loud. Can you not do that right now? Thank you. And then for the heterozygotes, it would be 2 times P times Q. And then for the homozygous recessives, then it would be Q squared, right? So remember, even though you have those three girls, it's too loud, please. Too loud. Thank you. So then for the Hardy-Weinberg equations are P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1. Really, those equations in and of themselves don't do you much good. You have to know what P and Q mean, and then you have to know what P squared, 2PQ, and Q squared mean. So really, it's a matter of calculating those things and making sure you know how to do them. If you were asked if a population was in Hardy-Weinberg equilibrium, of course, you could just look at them and see if they are close enough or not. But if you're really not sure, then remember with chi-squared, then you have to actually convert the percentages to numbers of individuals. So you should never do chi-squared with percentages like frequencies. You would only do it with numbers of individuals. So like with our lab data, right, we said like, 45% of individuals in North America were non-tasters. You would never use the 45%. You would say, okay, well, how, if I'm expecting 45% of like 50 or 45% of 100 or whatever it is, then you calculate the number of individuals that would be. So make sure for chi-squared, you're always doing numbers of individuals. Questions? Feel free to ask questions. Like I said, I don't know what you guys aren't sure about, so I'm just going to ramble on here. Let's see. Let me take a look at the practice quiz again then and see what else I can give you some tips on.
Well, that goes in our room too.
With evolution, really, there's a lot of vocabulary. So you want to make sure that you don't just know the vocabulary. You want to make sure you can also apply the vocabulary. So given examples of it and things like that, you want to be able to say, like identify either like what's going on or which term it applies to or doesn't apply to and things like that. Let's see. With the problem set, which I know you don't have right now because you turned in for 6A, keep in mind also a lot of that had to do with like assuming Hardy-Weinberg to be true and things like that, but also like being able to predict like how a population would change and things like that. Sorry, there's a lot of background noise here. Let's see. With natural selection, same thing. You want to be able to know sort of the idea behind how it happens and why it might change. You would have, again, you would want to be able to look at a scenario and figure out like either how natural selection would act on it or how natural selection has acted on it, or artificial selection and how that has acted on a population and such. Now remember, natural selection is just the idea that those individuals with the best adaptations to survive will survive and pass on their genes to the next generation. So over time, you're going to see that population change. But it is important to remember that, number one, they can't choose the ideal scenario of what they want, right? So you can't, generate a variation just because we really used to have it. You also would want to make sure that you're not, that there's no will involved, right? Like, again, the animals aren't saying we want to be this way, therefore we are this way. And then also just the idea that, you're going to see that population change gradually. It's not going to be in one generation. It's going to usually be very gradual as those changes kind of accumulate enough. And what can happen, but doesn't have to happen, is you could lead to new species forming, but it's not a guarantee, right? Just because a population changes does not mean it will become a species. So a new species. So remember that species are really defined by their reproductive isolation, by their inability to mate with other species, because in doing so now they are different, sufficiently different, whether it's because of their mating period or their habitat, or it could be other things like their mating calls and things like that. So again, there's a lot of vocabulary that you kind of want to be able to, again, don't memorize, but apply. Let's see. In terms of the PowerPoints, when you look in the website, if it helps you, then remember that there is one video lecture, and there's some other things on there. The videos, I don't know if they're actually still working or not. They keep changing the links, so I'll check those if they're not active. But you don't need the videos. Those were just for interest. Really. Thank you, Gina. Any questions you guys have? Like I said, just go ahead and put them up there, because I'm not sure what else you guys want me to talk about. Let's see.
Hello.
Sammy, they can't see you.
Hello.
Okay, Sammy.
Hello.
Okay, thank you.
I have a TV.
No, go, please. Go, please.
Mommy, I can't open this.
Okay, thank you. Okay, go, please. Let's see, other things I can talk about. I think that generally you want to make sure that you are able to connect topics too. I think the tendency with evolution sometimes is to put, certain terms all together and not necessarily try to connect them, but you do want to try to make sure you can connect the terms and see how they're related to each other, how you might be able to connect them, and things like that. Let me see. In terms of some of the later stuff we talked about with phylogenetic trees. With phylogenetic trees, you want to be able to read one and sort of see who's more closely related to whom. Remember, the later two things branch off from one another, the more closely related they are. Now, how you draw that tree really depends on your criteria, right? Because you could measure, you could look at like physical structures, or you could look at biochemical, like RNA, DNA sequences. So, how that tree gets drawn really might depend on that criteria. But the bigger idea is that you want to be able to read one. So I don't think the practice quiz has one of those. Let me check here. Yeah, it doesn't have one here, but again, the idea would be is you can, it's the simple idea that, you start with a common ancestor. As things branch off, then they become more different. That would be divergent evolution. And then Okay, girls, I really need you to be quieter. I don't think they can hear me over you. Okay, thank you. So then when you have things branch off, right? Again, the later they branch off from one another, the more closely related they are to one another. So do be able to read an evolutionary phylogenetic tree and, answer questions about it and that type of thing. That also, I think a common question I often get then is, do we need to know the scientists like Oprah and how they, for your quiz? No, I'm not really concerned that you know them. I would say for the AP exam, yes, you should know them just because they might bring them up. I don't think it's a major thing to know, but it is a small thing. Let's see. Let's see what else. Let's see. Adaptive radiation, gradualism, you should know those. You should definitely know your 5 kingdoms and your 3 domains as to whether or not, where something would fit and what the descriptions are. So on that note, 5th period, I know we never finished talking about I think the only one we hadn't done was the animals. So I will review those with you on Monday, but it's really a very, very tiny thing that you'll be fine getting on the last day. It's just the basic characteristics of animals, which you could probably already guess. They're eukaryotic, they are multicellular or single-celled. Or I think they're actually all multicellular. I don't think there are any single-celled animals. They're not photosynthetic, things like that. Okay. Questions? Anyone? Let's see. What else can I tell you about? So animals, multicellular heterotrophs, eukaryotes. Most are motile, but not all, and they don't have a cell wall. Let's see. Hopefully things are pretty self-explanatory in the PowerPoints, but again, if you need them, they're certainly there. Does anybody have any questions? Okay, so what would be helpful to you guys at this point? Do you want me to just kind of keep reviewing random topics? Do you want me to go over questions from the practice quiz? What will work? Anyone? Okay, thank you, Gina. Okay, thank you, Gina. Okay. I think there may have been something different, so I don't know if everyone's been asking questions. Oh, okay, sorry. Now I'm getting all these questions popping up. Okay, I'll pop a question or something. It says use your... Okay. Not sure what's going on here, but okay. Let me look up here. Okay, now I can suddenly see things that you guys are typing. So now I can see the comments. Yes, sorry. I couldn't see them before. Huh, sorry guys. I kept waiting for you guys to say something. Okay, yes, I can see these comments now. I'm not seeing comments from before though. Did you guys have comments from before? That's odd. Okay. So if anybody did have questions that I totally missed then, okay, thank you. Sorry. Sorry guys, I totally thought you were just like sitting there just watching me talk, which was awkward. Okay, so the difference between founder effect and geographic isolation, essentially they're related. Geographic isolation is what causes founder's effect. So founder's effect, remember, is the idea that when you have a group of organisms that leave the original population and they leave their original population and now go and sort of restart, right, a new population, if they were still able to be in contact with and mating with their original population, there really would be no way for them to change from the original population, right? But if they are now geographically isolated in such a way where they can no longer mate with their original population, then over time they will, not be so. They will not match the percentages and the allelic frequencies of the original, and so because of that, they will become different and they would evolve. So that's sort of founders. So you could say that geographic isolation causes founders effect. An easy way to remember the kingdoms and domains. Honestly, I don't know if there's a huge easy way to remember them. I mean, I would remember their names, obviously, animals, bacteria, etc. But I think if anything, I would kind of just try to remember key things about each of them and sort of the key mistakes. So, for example, like one of the big mistakes I see people make is saying that fungi are photosynthetic when they're not, right? So just remember, like, photosynthesis, no photosynthesis in fungi, like mushrooms don't photosynthesize. Domains, there's only 3 of them also. So just remember it's 2 different bacterial domains, bacteria and archaea. Bacteria are like your run-of-the-mill bacteria, whereas archaea are your like your more extreme bacteria, and then they lumped all the eukaryotes together in the eukarya domain. Okay, yes, you'll be given degrees of freedom. So for your chi-squared, if you need to calculate it, I will give you the equation. I will give you that table with the degrees of freedom and the critical values. You just need to be able to calculate the chi-squared and compare it to the critical value and know what to do. Let's see. Yes, so again, critical values will be in the table. That's a given. 2PQ stands for, so 2PQ stands for the expected percentage of heterozygotes, whereas so the reasons 2PQ instead of PQ is because, remember, like, let's just say like tasters and non-tasters, right? You have 2 alleles like big T, little t. It's 2PQ because you could be big T, little t, or little t, big T, and it's the same thing. So from like a mathematical standpoint, you have to like calculate both of them together to figure out the total number of people who would be heterozygous.
Nothing on the sofa.
Let's see.
Sounded like a mermaid.
Let me see if I missed anything.
The mermaid I have to take off? What?
Okay.
Mermaid.
Let's see. Other questions? Let me see if I answered everything. Okay, the difference between mechanical and gametic isolation is essentially where the issue lies that prevents fertilization. Mechanical isolation is the idea that sometimes physical body parts just don't match up, right? And because of that, then the chances of 2 organisms mating becomes really, really low. What, Gina? It's in the fridge. Whereas gametic isolation is the idea that there are species-specific proteins on the surface of the egg that prevent the sperm from another species from fertilizing it. So the example I gave you in class, which I think is actually a real one, is you can't take a hamster sperm and try to fertilize a human egg because there is a layer around it of human-specific proteins. So because the hamster doesn't have the right proteins, they won't be able to match up. So it can't even fertilize it. Like, it just can't even get in. Okay, the endosymbiont hypothesis is the idea of how eukaryotic organelles form. Specifically, it's how the mitochondria and the chloroplasts are thought to have formed. And basically what they think is it's sort of like Pac-Man, that one prokaryote essentially engulfed or ate another one, and then that prokaryote that got eaten was the one that became the new organelle. So like mitochondria have their own DNA, which suggests they at one time may have been a whole separate entity, right? So the thought is that they got eaten and became, an organelle within another prokaryote, which is sort of now how you started to have eukaryotes that have sort of organelles. And then the same thing happened, but with another one, which is photosynthetic, and that's how chloroplasts came to be. Yes, I can go over the Haldane hypothesis and the Miller-Urey experiment. So basically, this is one of the most famous experiments of how they think life may have originated from abiotic or non-living factors, right? And so Oberon and Haldane thought that based on their evidence, there was essentially all these gases in the atmosphere, but no oxygen. So you had methane, you had ammonia, you had hydrogen gas, and then you had a lot of ocean water, and it was really, really warm. So then the water would heat up because of that high temperature, and water vapor would now enter the atmosphere as well, right? So now you had water vapor, gaseous water, in the atmosphere as well as those gases. When lightning struck and provided electricity and electrons, then that would basically cause these atoms to sort of be able to combine and rearrange in such a way where then Oberon and Haldane said you would form fatty acids and amino acids. What Miller and Urey did was they basically said, well, let's test Oberon and Haldane's hypothesis. So that's the picture in your notes where they created an experiment to simulate the conditions that that Oberon and Haldane said were present. And then when they sort of did the whole process of you had the oceans and the water, and no, you don't need to know these scientists in detail. I would just know the general idea of what they thought. Right. When they did that, they were able to see that they did actually generate amino acids in their solution. So that told them that, yes, the hypothesis was very plausible. Okay, what's the difference between reduced hybrid viability and reduced hybrid fertility? So both have to do with if sperm and egg are successful in actually producing offspring, just because of that doesn't mean now you have a new species, right? It means that in order to really be a new species, it has to be like you can keep carrying on that family line all the way down. So reduced hybrid viability is the idea that you can have an offspring, and when you have the offspring, it's actually kind of weaker or not able to develop as well. And so because of that, then you end up with not a sustainable line in the family tree. And then a reduced hybrid fertility is the idea that sometimes those animals are perfectly strong, but instead their issue is that they cannot have children of their own. So the family line pretty much stops there, right? So you have like mules are a great example because they're really, really strong animals. They don't, they're not weak or underdeveloped at all. They're using a lot of farm work. But now what you find is that they are sterile. So you can't have two mules have a baby mule, right? And so because of that, it's not a new species because it just ends there. You have horse and donkey give birth to a mule, and that's the end of the line. There's no more passing down of genes, etc. The gene pool essentially is done at that point for that, that organism. They're not going to contribute. Phenotypic polymorphism is just a very fancy way of saying that one of the things we talk about in evolution is in order to have populations change, right, you have to have variety in the population so that somebody can do better than somebody else. And so phenotypic polymorphism is just a fancy way of saying having a variety of physical traits in the population. A lot of that is due to genetics, right? Like different alleles, the fact that an individual has multiple alleles that they can then pass down to their children, but then also that you have more than usually one allele that exists, right? So there's a variety that can be present. So really that's all you need to know about phenotypic polymorphism. It's just the idea of like you have a variety of traits, and that's either due to both, it's due to a combination of genetic and non-heritable, aspects. Let's see. Other questions? Let's see. Okay. So really, it's a very subtle difference. So cells working as a colony means like they're all still individual organisms, but they kind of work together on shared tasks, right? So like a group of protists, like each protist is still an individual organism, but they might be doing certain shared tasks that all contribute to their well-being. Whereas cells working in a multicellular organism, like I can't take your stomach cell and like put it outside and have it function all by itself, right? It's part of your whole body. So it's an individual cell, but it's part of a larger multicellular organism that it's functioning as. So it can't function. Your stomach cell cannot function all by itself outside of your body. Okay, yes, the RNA world hypothesis is just the idea that, we're really used to DNA being sort of the big genetic molecule we hear about all the time. And there is a sort of I wouldn't say competing hypothesis, but just sort of like an alternate hypothesis that says maybe RNA actually came before DNA. And that because of that, we see RNA having certain traits that suggest sort of that it may have another time done more than it does now today, right? Because today we're used to it sort of just being the in-between molecule, the one that helps us go from DNA to protein, the one that helps us make proteins. But they've actually found a lot of other things that RNA seems to do. Things like it seems actually some RNAs, like ribozymes, seem to have certain enzymatic qualities. Like they used to think that in your ribosomes, the main thing that was doing the catalyzing was the protein. But then they actually found out that there were some RNAs in there that were actually doing the catalyzing. And because of that, then again, it helps suggest that maybe, yes, at one point, RNA was sort of the main genetic molecule, and then it may have actually done some of the things that DNA and proteins do now, prior to sort of DNA and proteins developing, etc. Let's see. Other questions? Let's see. Let me see what else can I look at for you all. Other questions? Let's see. Let me see and make sure I've gotten all the questions that have been asked so far. Let's see. I guess something to also remind you guys of really quickly is that for chi-squared, we use it in the context of Hardy-Weinberg, but I do want to sort of make sure you are reminded that chi-squared doesn't have to be for Hardy-Weinberg. We use it for Hardy-Weinberg because it is one way to figure out if what we thought we were going to get was the same as what we actually got. But just keep in mind that Hardy-Weinberg is only, or applying it to Hardy-Weinberg is only one way that chi-squared is used. Chi-squared is really something you can use anytime you want to find out if what you thought you were going to get is close enough to what you actually got. So you could do it for, it's very commonly done in like experiments then to see like if my hypothesized results match my actual results and things like that. Because I do think people tend to assume, because we use it only for Hardy-Weinberg, that that's the only thing we use it for, but it's not. Definitely, is a very generic way to just see if what I thought I was going to get is what I actually got. Okay, let's see. Let's see. This is not the longest unit, huh? So there's not as much stuff to ask maybe either. A practice chi-squared. Yes, I could. So there's a couple. So maybe a one way for us to talk about chi-squared is, the only time you would do chi-squared is if you're trying to figure out if what you thought you were going to get was what you actually got, right? So in this case, Tajin, you said something about I'm not sure when to take the square root of values or insert them into the Hardy-Weinberg equation. I would have to tell you either like there's two different scenarios. I could either say assume the population is in Hardy-Weinberg equilibrium, which means you have to apply the equations, right? Or if you're looking at a scenario where you're trying to figure out if it is or is not in Hardy-Weinberg equilibrium, then you do chi-squared. In which case you would take your observed values, which is usually the data you've collected, and you'd compare that to expected values, which is going to be then where you apply the Hardy-Weinberg equation. So in that case, you would calculate P and Q first, right? Now you have your allelic frequencies. And what Hardy-Weinberg said was if I know my allelic frequencies, I can predict what my genotype frequency should be. So then you do P squared, 2PQ, and Q squared. In doing that, you get expected genotype frequencies. If you have data, then you're going to compare that to your actual genotype frequencies or genotype number of, that have each genotype. So if you're wanting now to figure out if it's close enough or not, then you would have to take your expected genotype frequencies, multiply that by the total number of individuals in your actual population to figure out how many people you would have expected to have that. And then you do the chi-squared comparison between actual number of individuals and expected numbers of individuals based on that. I don't know if that helps or not. I can try to find a question. Maybe I'll take a look in a second at the practice quiz. There was a question about what's the difference between convergent and divergent evolution. Convergent and divergent evolution are very different, right? Convergent evolution is the idea of like bird wings and butterfly wings, the idea that you can have organisms evolve similarly, even though they don't have a common ancestor, because they have similar environmental pressures, selective pressures, right? Whereas divergent evolution is the idea of like you have a common ancestor and then they branch off into different branches and could end up being quite a bit different, but we see evidence of their common ancestry through things like physical structures or other things like that. And maybe another way of thinking about it is analogous structures would be formed through convergent evolution, whereas homologous structures, where today they probably look and do very different things, but still have hints of the original, similarities, would be formed through divergent evolution. Another term you can associate with divergent evolution would be adaptive radiation, right? Because the idea that you start from a common ancestor and then you branch off into lots of different potential species from there. Let's see. So let me take a look at the quiz for a second. And since there was a question about chi-squared, I'll just kind of walk you through one really quickly, especially since the quiz has a lot of chi-squared practices. So maybe one I can use, let's see. So if I look at number 4, right, which has to do with Daphnia, I have, it's for albinism, right? Big A is a dominant or dark color. Little a means no color. And I have the number. So all those numbers there, like 846, 148, and 6, those are the actual numbers of individuals. So that means that's going to be the stuff I use for my actual or observed frequencies, right? In this case, the number of total alleles, I would have to add up all the Daphnia, and then I would multiply that by 2, right? For allelic frequency, then I would calculate it just by doing what I did on the very first day, right? So I take if it was for big A, it's going to be 846 times 2 plus 148 divided by my total. Now, genotype frequency of little a, again, is 6 over the total. So all of that is just straight calculation. In part D, notice now it says using the Hardy-Weinberg equation. Now, I may or may not always say that. I might just say, what is the expected genotype frequency? So anytime you see expected, then you should say, well, that's assuming Hardy-Weinberg, right? So now you're going to say, well, I already know P, which is the answer from B. And then I can find Q from taking 1 minus that, right? And then I can figure out the expected genotype frequency of homozygous recessives would be Q squared. Now, for part E, where it says, is this Daphnia population in Hardy-Weinberg equilibrium? this is where you got to do the work, right? Where first you would calculate p squared, 2pq, and q squared. Those are my expected genotype frequencies. I would calculate my actual genotype frequencies, which would be the 846 over the total 140 what is it, 148 over the total, etc., right? And then the key here is if I'm going to do chi-squared, and again you would get that, then I have to convert those p squared, 2pq, and q squared percentages into out of the total, which looks like 1,000 Daphnia, right? So out of 1,000, like how many do I expect? And then I take that expected number and compare it to 846, 148, and 6. Okay. Other questions? Let's see. Let me take a look at the problem set too. Yeah, the problem set really is largely, mostly hard to remember practice. So just, again, don't be misled in thinking there's going to be that much of it on your quiz. There is some, but not that much. Other questions? Let's see what other practice I can find for you all here. I would say definitely make sure that you understand, again, the different types of selection and being able to apply that. Yes, I can go over adaptive radiation. Adaptive radiation is the idea that you start with one common ancestor, and because there ends up being a fairly large diversification of, let's say, environments or environmental conditions, then that one original organism can branch off or diverge into lots of different species. And so, it's something that you can sort of link to things like homologous structures, but also like just the idea that you can have things branch out when the assumption would be that there have to be now lots of different, very distinct environments such that organisms, evolve to be quite different. And so the example I gave you in class was the Hawaiian honeycreeper birds, where they think the American finch first came to those Hawaiian Islands, but because they now found all these little niches and all these little pockets in different environments to survive in, they eventually diverged to be about 23 different Hawaiian honeycreeper bird species. Let's see. No problem. Let's see. What else? I would say also for like just general evolution, make sure you know, like I said, all the different terms, right? So things like gene flow, but also like heterozygote advantage. Remember allopatric versus sympatric speciation. I think I gave you ways to remember that, but in case you still need that, remember like allopatric means it's due to geographic isolation. So I always think allo alone, right? They're alone, they're geographically isolated. Whereas sympatric speciation means that you have some other reason why the species become distinct from one another, but it's not because of geographic isolation. It could be mutations, it could be polyploidy, things like that. So as a result, they are in a similar environment, so similar, sympatric, etc. Let's see. Let's see. Oh, fitness. Make sure you know about fitness and sort of just the idea of fitness, right? In this case, it doesn't mean anything about how many sit ups you can do. It's really, in this case, about evolutionary fitness is about reproduction. So, an organism who reproduces and contributes to the gene pool more is considered to have higher fitness, regardless of how physically strong he or she is. Let's see.
Just to make double, triple sure. I'll get it.
Um,
Bring on, Mama.
let's see. Sorry, I'm just trying to make sure I cover stuff for you guys because I'm not sure what else would be useful at this point.
Carnival.
Do you want me to go over polyploidy or is that okay? Want to know things like your post-zygotic, pre-zygotic berries. Okay, so let's go over polyploidy because I think that one can be a little confusing. Polyploidy is just one way that specifically plants can essentially have speciation occur, right? I mean, at the core, polyploidy is defined as just having too many chromosomes. And normally we just think of that as like it's a bad thing and nothing happens, but like you can't have new species result. But in plants, what can happen if there's enough sort of pieces to that, then you can basically end up with new plants that are able to have children of their own, but are now reproductively isolated from their parents because they have the wrong number of chromosomes. So it's usually due to some mistake or multiple mistakes in cell division, such that you end up with gametes that have too many chromosomes, but they're still able to pair up in a certain way such that they can still, like I said, go through meiosis, still make gametes that are able to, reproduce. And in the case of plants can technically self-fertilize. So that means even if you have only one plant that had this issue, like theoretically, it can still, like reproduce because it can, without getting too much into it, because we haven't gotten into plants too much, there are like male and female parts to plants. And so it's possible for those gametes to mix and sort of now generate another plant and sort of just propagate that in that way. And like I said, it's, your pictures, the pictures in your notes will give you specifics. That's the idea. Endosymbiosis is the theory of how mitochondria and chloroplasts came to be organelles. So essentially, it's thought that one prokaryotic organism essentially ate another one, kind of like Pac-Man, and the one that got eaten became the chloroplast and the mitochondria. And the evidence to support that is that there is DNA, separate DNA in these organelles, right? So it suggests that at one point in time, this this organelle was separate. Epistasis and dominance. Okay, that's actually not on the quiz, but I'll talk about it anyways. So epistasis, this goes back to genetics, right? Epistasis is the idea that one gene is controlling another gene. So like whether or not a mouse is able to show what color it is controlled by another gene, then that's epistasis, right? Whereas dominance is just the idea that you only need one copy of said gene to show the trait. But yeah, that's not on your quiz, so don't worry. Is it somehow in your quiz? Like, is it on the practice quiz? Hopefully not, because again, you don't need to know it.
Because this is the first day she got that.
Just mention 10F. Okay. Let me see.
Snack, Mommy.
Go ahead. Oh, I see. It's just an answer. Okay. Yeah. So, I mean, you don't have to know. Okay.
Can I see me?
Mhm. Okay, can you go get a tissue for yourself, please?
Okay, let me just read the question.
Okay. Go get a tissue. Okay, thank you. Go get a tissue. Sniffles are not useful to you. No, thank you.
Bobby, can I send my Girl Scout cookies to you today?
Yes, but not right now.
Okay.
Go get your tissue, please.
Okay. Can I just show you
Can you
one
go?
box
Not right now.
really quick?
Okay, you go to his video. Thank you. Okay, let's see. No. Tissue first, please. Tissue before Girl Scout cookies. Thank you.
Where's the tissues?
Tissues are right there.
Let's
Okay, maybe it's not there. It's okay. Calm down.
Can I please have 3?
Go ask Daddy.
No,
Go ask Daddy.
Sammy's gonna open the door.
It's okay. It's okay. Just close the door. Gina, there's probably tissue back there too. Okay. You just have to wait for mommy then. Okay. Yeah, you can too.
What?
Let's see.
Since you're eating this. No, I'm not. I'm showing mommy. Not eating it. I still got
Make sure you know about your post-zygotic and pre-zygotic barriers, right? Understand sort of what type of barrier something would be qualified as and why. In both cases, they just come back to the idea that they both somehow prevent that organism from being able to have like a sustained, line, right? That to sort of have a whole new branch off a family tree. Okay. Let's see. What else? I guess another thing I can sort of briefly talk about is just remember sort of the idea that when we have evolution is defined as change in a population, right? And so that can be caused by a variety of things. Go get a tissue, please.
Okay, I can't find
Okay, go put your Santa socks on then because you're cold. Okay. A primitive and derived trait, I didn't really talk about that too much, so I really wouldn't worry about that. I believe a primitive trait is just something that's sort of, there present at a very early common ancestor, whereas derived, I think, means it develops through that branching. Okay, thank you, Sammy. But yeah, I wouldn't worry about those terms. We didn't talk about them at all, so I don't expect you to know them. Okay, let's see. Also remember the idea of sexual selection, right? The idea that you can have, males and females look different from one another, and because of that, often it's because then the males are competing for the attention of females. Okay, hold on, girls. AP test prep, we're actually going to start really soon. I don't know if you've noticed, we have these big stacks of paper in the room right now. And so just sort of getting us through this quiz first, and then I'm going to introduce the AP prep to you probably next week, like this coming week. I just have to figure out the day that it works best because we're a little tight on time. But basically, once we start AP prep, then we'll do like this 12-week review while we're still covering new content. And we'll do that. And then I have these online videos that you'll be able to watch to help you review the content as well. And then as we get closer to the AP exam, I'll have like mock APs that you can do and try. We'll do some in class. I'll have some after school, and some will just be available to you, kind of like the one you did last Friday when I was gone. But the idea is sort of to gradually, no, thank you, Sammy, to gradually sort of give you more and more familiarity with sort of how they answer questions. And then
is Kina watching me?
No, this is not her class.
This is
these are the older kids that you guys they in the afternoon. So
have I been?
Yes.
Oh, I've seen them.
So phenotypic and genetic polymorphism is just polymorphism really just means many forms, right? So phenotypic polymorphism is the idea that you're going to have lots of different physical variations, like lots of different phenotypes in the population. A genetic polymorphism is the idea that you're going to have different forms genetically, so like different alleles.
Sammy, you forgot me.
And so, I mean, ultimately, they both lead to variation in the population, right? But it's just sort of the more particular aspect of like what's causing or what type of variation is that. Is it a genetic polymorphism, which again would be like alleles, or a phenotypic polymorphism, which would be like just a variety of physical traits, which can be then in many cases derived from or caused by genetic polymorphism, but doesn't have to be. Okay, let's see what else. Let's see. Again, make sure you can identify which kingdom, which domain something is in. Yeah, Sammy. You need to wait till mommy's done in a few minutes. No, not yet. You need to wait. You need to wait. If you pick it now, then I'll talk to you when I'm done. Okay? And you may not have it till I'm done. Thank you.
Until mommy's still talking.
Yes, mommy's still talking.
Can I pick right now?
You may pick right now, but you may not eat right now. Thank you.
Me too.
Do you want you might also want to take a look at those last few slides on the evolution PowerPoint, the ones that have to do with like constraints on natural selection. Just a good reminder of, even though we talk about natural selection might be acting on a population, that there are constraints to it. Okay, yes. Concept of booming diversity. Booming diversity is just the idea that you're going to see times when there's more speciation happening than others. And that's usually when there's some sort of like opportunity or event happens in the environment, such that now all of a sudden you have these girls. Can you go over there, please? Now that you have sort of opportunities for organisms to be able to, fill niches and things like that. So, for example, like right after the dinosaurs went extinct, you saw a lot more mammal species coming to be because now they could take over the niches that the dinosaurs filled. Sexual dimorphism is the idea that in a lot of populations, males and females look different. No, thank you. Sexual dimorphism, no, thank you, Sammy. It's just the idea that you have, you're going to have differences between the males and females. And as a result of that, usually it's what drives, no, thank you, Sammy. No, thank you. It drives the competition, right, in the population, because now if you have, like, for example, peacocks with like different sized tails, then they're competing with the other peacocks for the attention of the females. So it's a way in which it would drive evolution of the population. Well, you have to wait till I'm done.
You have to wait until she's all done talking.
You're welcome.
What? But you're welcome.
They said thank you, so I'm saying you're welcome. It looks like we're almost out of time here. Does anybody have any last questions? I'm so sorry I couldn't see them earlier. I don't know why it didn't default to that window. But I will still be available tomorrow at school if you guys have any questions.
I'm gonna miss you tomorrow.
Okay.
Mommy,
It's not
tomorrow can I
yet.
sell small cookies to Auntie McKenna, who
Yes,
is
you
Miss
can.
Parfait?
Oh, Gina, you have yes. Gina will be happy to sell you Girl Scout cookies.
Wait, what? Yahoo!
Okay.
I'm selling Girl Scout cookies at your
Actually, let me
school.
double check. I got to find out when she's allowed to start selling. But as soon as she's allowed to start selling, yes, we'll We'll
No, I
ask.
can start tomorrow.
You can start tomorrow? Okay.
I can only sell it to you guys today.
Oh, okay.
And maybe Mr. Peter
Okay.
across the street.
Oh,
How about
I'll-
the other neighbors next door?
we'll
to
give you
us?
cookie information about our question.
Mommy, can I eat this?
No, I'm not done yet, sweetie.
But,
Yes, I think she has everything.
but it
We've got-
already show you one really quick.
Oh my goodness. No, we're not supposed to be selling girls. No, you can't sell today. Remember? No, I don't think it's 7. I think it's 5. I don't know. Do prices go up?
Showing them one
Gina, how much is one box of cookies?
$5
Oh, $5.
or sometimes it's $6.
Okay.
And Evelyn said to be generous anyway.
Oh, okay. Yeah.
January.
Okay, there we go. Thank you, Gina.
Can I eat this?
Well, let me say goodbye to the students, and then when I'm done, you may say you may eat it. Okay. All right. So it sounds we've transitioned. It sounds like away from evolution.
What? Why are you laughing at?
It's okay. Anyways.
Bye-bye.
Yeah.
I hope. What is that? Why
Nothing.
do they say that?
Okay. No
What?
problem, guys.
Why do they say that?
Hold on. Okay, hold on. Everyone say bye-bye.
Bye-bye. Bye-bye.
Okay. All right. No problem, guys. I'll see you guys tomorrow. Let me know if you've got questions, and have a good rest of your weekend, okay? All right. Bye-bye.
You can open this. Can you help me?
Can you wait?